Hello, everyone. My name is Yang Shen, and I'm a scientist in the data science and bioinformatics team at Crown Bioscience. Thank you for joining this webinar. The topic of my presentation today is impact of RNA degradation on transcriptomic profiling. In particular, I'll be focusing on high-throughput next-generation RNA sequencing, which is currently the most widely used technique for measuring the expression levels of transcripts in cells and tissues. A typical RNA-seq experiment consists of isolating RNA, converting it to cDNA, preparing the sequencing library, and sequencing it on an NGS platform. RNA-seq works under the assumption that every nucleotide of the transcript has an equal chance to be sequenced, and the number of reads produced from a transcript is proportional to the abundance and length of the transcript. So when RNA molecules are partially or completely degraded, the corresponding read yield would also be distorted. Therefore, RNA-seq requires high-quality RNA samples in order to produce reliable and reproducible results. Checking for RNA degradation has always been an important part of quality control in the RNA-seq workflow. A commonly adopted measure of RNA integrity is Agilent's RING, which stands for RNA integrity number. This method uses a machine learning approach to extract features from bioanalyzer traces and train a neural network that is predictive of RNA integrity in a large set of samples manually curated by experts. Ring value can range from 1 to 10. By design, the higher the ring value, the better the RNA quality is supposed to be. Incidentally, the highest ranking features in the neural network model turn out to be those that heavily relied on the amount of 18S and 28S ribosomal RNAs. It follows then as a reasonable conjecture that the ring metric mainly captures the integrity of ribosomal RNAs and does not directly reflect the integrity of messenger RNAs. This is considered to be a limitation because, after all, most RNA-seq experiments are concerned with measuring the abundance of transcripts. On the other hand, once an RNA sample has been sequenced, downstream analysis of the sequencing data can provide additional information about the quality of the starting sample. For instance, for RNA-seq libraries prepared by poly-A selection, when the starting RNA sample is heavily degraded, the distribution of the sequencing reads tends to be biased towards the 3' end. This can be seen in this graph on the right, where read coverage averaged across all transcripts is highly skewed in these samples with low RNA quality, but shows a more even pattern for these samples with high RNA quality. Based on this phenomenon, the transcript integrity number, or TIN, has been proposed as an alternative quality measure to RING. For each individual transcript, TIN uses an algorithm based on Shannon's entropy to quantify how uniform the sequencing reads are distributed along the length of the transcript. In theory, the value of TIN can range from 0 to 100, with 100 representing perfectly uniform read coverage and 0 the opposite. In practice, though, since perfectly uniform read coverage is not possible, even for samples with high quality, the value of TIN never really reaches 100. We can then take the median of the TIN scores of all the transcripts and use the median TIN score as a metric to measure the overall RNA integrity of a sample. So to summarize what I've told you so far, when we compare and contrast these two RNA quality metrics, namely RING and median TIN score, we can see that they are different both in terms of the kind of information they're taking and the aspects of RNA sample quality that they reflect. Some has argued that compared to RING, the median TIN score is a more relevant quality metric for RNA-seq experiments because it more directly measures the integrity of transcripts. The nice thing about RING, though, is that it doesn't require read alignment and allows us to exclude low-quality samples before sequencing. Typically, RNA samples with ring larger than 7 are considered high quality and suitable for library construction by poly-A capture. Of course, then, when we exclude RNA samples with a low ring value, we are always implicitly assuming that the extent of degradation for ribosomal RNAs and that for messenger RNAs are positively correlated. But is this actually the case? In the next 10 or so minutes, I'm going to share with you a case study that we did using a bulk RNA-seq dataset of syngeneic tumor models. These are allograft tumor models derived by engrafting mouse cancer cell lines into immunocompetent mouse strains with matched genetic backgrounds. The sequencing library was prepared by poly-A capture, which enriches for polyadenylated messenger RNAs. This dataset consists of RNA samples with a wide range of ring values, making it ideal for answering the question that I posed just now. When ring values and median t-scores are plotted together, we did see a moderate positive correlation, which is statistically significant. Interestingly, however, when we plotted these two quality metrics against each other separately for different syngeneic models, we saw two strikingly different patterns in the data. For model types that are labeled in black, there is a high concordance between ring and median t-score, which is evident from the data points lying close to the regression line and the high correlation coefficients. It means that for these model types, using median t-score and ring to evaluate sample RNA quality will largely yield consistent results, which then support to the practice of using ring as a pre-sequencing quality check metric. But the same cannot be said for all model types because for syngeneic models labeled in red, the correlation coefficients are close to 0. For such sample types, for example, in EL4, if we use a cutoff of ring larger than 7, we run the risk of discarding samples with actually pretty high transcript integrity that is comparable to those samples that are retained. This would have created a huge waste if we were dealing with clinical samples that are hard to procure. We also noticed one CT26 sample with a high ring value, but an extremely low median t-score. Further analysis, which I'm going to show later, confirmed that this sample was actually of low transcript integrity, which would not have been detected if we used ring as the only metric for sample RNA quality. So taken together, our analysis so far has demonstrated that the intactness of transcripts and ribosomal RNAs are not always positively correlated. A tentative interpretation here is that depending on the tissue or cell type, it is possible for an RNA sample to have heavily degraded ribosomal RNAs while still retaining relatively intact messenger RNAs. Since ring and median t-score each provide complementary information about different aspects of sample RNA quality, we should consider using both in the quality control of RNA-seq experiments. Although excluding low-quality samples certainly works when the experiment can be easily repeated, we are aware that there might be situations where the samples of interest are extremely hard to come by, and one has to work with RNA samples that have undergone some form of degradation. For that reason, we also wanted to find out to what extent RNA degradation would impact the results of downstream analysis. When we perform principal component analysis on transcript-level read count data, we found that the first principal component appears to be representative of the true biological differences in the expression profiles of different syngeneic models, because the distribution of data points in this direction are nicely separated by model types and show no correlation with either ring or median t-score. The second principal component, or PC2, on the other hand, shows a strong negative correlation with ring and also appears to decrease monotonically with increasing median t-scores. In other words, the observed expression levels for genes with large loadings in PC2 are heavily biased by the presence of RNA degradation. So in a sense, we can look at PC2 as a proxy for the degree of RNA degradation. And returning to a point that I made earlier, notice that there is this outlying CT26 sample with decent ring value, but actually the smallest median t-score among all samples. The fact that its location sort of deviates from the other samples in the PC2 versus ring plot, but appears to be consistent with the overall decreasing trend in the median t versus PC2 plot, supports the idea that transcript integrity is actually very low in this sample, which is not correctly reflected by its inflated ring value. If we summarize the expression data from all transcripts into a gene-level read count matrix and perform PCA, then in stark contrast to what we saw earlier, samples from the same model types now cluster together very nicely. When plotting PC1 and PC2 against ring and median t, we now see these parallel stripes that are suggestive of zero or very weak correlation between the expression values of the genes represented by these two principal components and the extent of RNA degradation in the sample. By comparing the results from visualizing transcript level and gene level expression data, we can conclude that the impact of RNA degradation on gene level data is much smaller than on the transcript level expression data. So intuitively, one would hypothesize that an analysis performed on gene-level expression data would be more reliable than a downstream analysis on transcript-level data, since the former is less distorted by the presence of possible RNA degradation. To test this idea, we performed both gene-level and transcript-level differential expression analysis on KLN205 and EG70VA samples. First, we define a group of high-quality samples as a standard set. These are samples with both ring larger than or equal to 7 and median t-score larger than or equal to 70. This standard set represents an ideal situation where all samples in the differential expression analysis have passed a very stringent quality control criterion. The results we get from this standard set are treated as the quote-unquote correct answer. Then to mimic the situation where samples of suboptimal RNA quality are included in the analysis, for the test sets, we randomly take the same number of samples as the standard set from all samples regardless of their ring and median t values. After repeating this procedure for 10 times, we then compare the results that we got from 10 randomly sampled test sets with the quote-unquote correct answer that we got from the high-quality standard sets. Below on the left, the replication rate stands for the proportion of differentially expressed genes that were identified in the standard sets and successfully replicated in the test sets. False discovery rate stands for the proportion of differentially expressed genes that were identified in the test set but not in the standard set, so they are considered false discovery. Obviously, the higher the replication rates and the lower the false discovery rates, the better, meaning that the impact of RNA degradation on the result of the analysis is smaller. And just as expected, we see that the gene-level analysis represented by these red data points always shows higher replication rate than the transcript level analysis represented by these blue data points, and also lower false discovery rates, supporting the idea that gene-level analysis more robust. And we also found that this is true when we vary the cutoffs for differentially expressed genes, both in terms of the adjusted p-values and in terms of the absolute log-to fold change. Now, just a quick summary of the two take-home messages from my presentation. First, ring value alone does not provide a complete picture of sample RNA quality, and we suggest using both ring and median t-score in the quality control process of RNA-seq experiments. Second, we saw from our analysis that when samples of suboptimal RNA quality are included in RNA-seq experiments, gene-level differential expression analysis are more robust than transcript-level differential expression analysis. Thank you again for joining this webinar. If you have any questions or need further information, please feel free to ask now or reach out to us later via email.